span the years 2016 to 2022, and from the newly initiated Aging Adult Brain Connectome Study, which will begin enrollment in the coming months. Given the public availability of the HCP-A data and the planned availability of the AABC data, we believe this session should be a useful introduction to these projects and their accompanying resources. There will be 4 presentations in this symposium. The first presentation will be by Dr. Bo Ansis and is entitled Vulnerability to and Resilience Against Risk Factors and Biomarkers of Alzheimer's Disease. In this presentation, Dr. Ansis will describe the projects and cores of the AABC project and how this design allows investigation into factors that contribute to cognitive risk as well as protection against cognitive and neural decline in late life. The second presentation will be done remotely by video by Dr. Matthew Glasser, entitled The HCP-Style Approach to Brain Imaging and Imaging Data Phenotypes, in which he will describe the imaging methodology that is central to the HCP-A and AABC projects. The third presentation will be by Dr. Bornali Rashid and is entitled Early Risk and Late Life Resilience, in which she will describe an examination of the impact of cardiometabolic health on brain functional connectivity in the HCP-A AABC cohort. Dr. Melissa Terpstra will give the final presentation entitled Neurochemical Profiles of Late Life Cognitive Risk, in which she'll describe novel magnetic resonance spectroscopy applications in an ancillary study to HCP-A, which led to the addition of 7 Tesla magnetic resonance spectroscopy acquisitions for the AABC project. All of the presentations you will hear in this session would not be possible without the contributions from a great number of individuals included on this slide, as well as others not listed here. The HCP lifespan studies, which include partially harmonized aging and development data sets, are described in these 3 neuroimage publications listed here. We would like to particularly note the central leadership of Drs. David Van Essen and Deanna Barch at Washington University, and the aging site principal investigators and core contributors, including Dr. Susan Buchheimer and Roger Woods at UCLA, Dr. Melissa Terpstra at University of Minnesota, now at University of Missouri, Dr. Issa Yacoub and Camille Ugerbel at University of Minnesota, Drs. Bo Ansis, Michael Harms, and Matthew Glasser at Washington University, and Dr. Randy Buckner at Massachusetts General Hospital, Harvard University, and Dr. Stephen Smith and colleagues at Oxford University. For the AABC extension to HCP-A, we were joined by a number of new investigators listed at the bottom right of this slide, who have made substantial contributions to the design and execution of this new phase of the project. We would like to particularly note the contributions of Angela Oliver, the project manager for AABC, who has put great effort towards startup operations for this AABC new phase. Additionally, we'd like to acknowledge the support from the National Institutes of Health and National Institutes on Aging for HCP-A and AABC through the U01 and U19 projects listed here, as well as the National Institutes of Mental Health Support of Human Connectome Project Development through the U01 project listed here. And Dr. Terpstra's ancillary magnetic resonance spectroscopy work was also supported through an R01 from NIA. We'll now begin the session with Dr. Ansis. Thank you.
So welcome, everybody. This is the first talk, and we're so honored to be starting off OHBM. It's great to see everybody here and those that are online, and especially if you're in the States, God knows what time of the morning it is. I'm Bo Ansis, and we'll be talking about the adult aging brain connectome, vulnerability and risk against and biomarkers for Alzheimer's disease. Here are my disclosures. The points that you already saw David already talk about. The one in red is our new U19 that talks about the AABC. Of note, I have no stocks or equity in any pharmaceutical companies. What we're going to be talking about today is first the brief history of the adult aging brain connectome. We'll be talking about hypotheses that are guiding the AABC, understanding these trajectories of what is healthy aging versus what is disease. We'll also be talking about projects and cores in AABC and the new leaders and existing leaders for these projects, and then what variables that will we be collecting and what will be made available to everyone, and then even some very interesting preliminary results. And as already heard, Drs. Terpstra and Rashid will also be presenting some of those very interesting results. So really, this is the transition from the Human Connectome Human Connectome projects to now the AABC. So originally, as many of you know, there was the original HCP young adult study. It was from 2010 to 16 that studied 1,200 healthy adults. It was acquired at multiple sites. That then led to the lifespans, the HCPA studies of about 1,200 older adults, as you can see from their ages, from 4 different sites, including WashU, Minnesota, UCLA, and Harvard MGH. In many of these cases, there were both longitudinal scans as well as there was also some individuals had a single scan. The scanning was about 90 minutes in duration, and then the first release, as many of you know, under the NIMH data archives was done in 2019 with more soon to come. That really spurned a whole group of other studies that were going on, including the baby connectome projects and then multiple connectomes in disease. And now really what we're seeing now, and that we've been very fortunate enough, and I'm proud to announce we already had our first participant enrolled and studied as of a couple weeks ago, the AABC, which is a new extension and iteration of this HCPA with a couple little twists, and I hope that will make it even more available to the population. So the AABC, as you can see, builds upon existing cohorts, so there'll be around, of the 580 cross-sectional time points, we'll be studying those individuals 2 more times, so that'll be 4 potential time points in some individuals. In some individuals, they've had 1 single time point, and that'll allow us to have about 350 individuals with 3 time points in total, because at the AABC, every individual will have 2 scans. And then some newer individuals that will be brought in, cohort C, as you can see, will have 2 time points. So the idea will be is that in many cases, we will have up to 4 time points over more than 10 years of chronological age for these individuals. Now, what are we really very interested? So we're really guided by 3 hypotheses. One is to look at the integrity of the connectome, both structurally and functionally, as it changes with healthy aging. And then the idea is that when there are disruptions in these connectomes, that is really an underlying mechanism for certain kinds of neurodegenerative diseases, the most common, of course, being Alzheimer's disease or AD-related dementias. Now, we're also going to be looking at multiple factors that could be, including genetic, behavioral, social, cultural, and environmental, and how that is either protective or deleterious, so resiliency versus vulnerability. And then we are very interested in these key transition points, meaning that there are particular phases during the lifespan that can lead to divergences in the trajectories. And in particular, we're looking at menopause, midlife, and late life. You can see on the right-hand side, we have the age and then the connectivity in both structural and functional. And you can see those declines as we see with aging. So I'm sorry, as we're sitting here right now, we're having some of those declines. But what we're seeing is that there are key kinds of points or phases during the aging process, and if individuals go off of those curves, why are they going off of those curves? And so the effects of certain factors across the entire lifespan will be determining an individual's late life risk for pathology. So what are the projects that are going to be incorporated into the AABC? Well, there's Project 1, which is the study of allostatic load and also immune dysfunction in early adult life. Project 2, which is really looking at lifestyle behaviors, in particular physical activity, sleep, and metabolic changes that can be occurring. Project 3 is to look at menopause and its effects both through vasomotor symptoms and other skin conductance measurements. And then Project 4 is to study resistance against AD. So who are the healthiest of the healthiest? How are they able to successfully age? And you can see that there are interconnections between those various projects as seen in the right-hand figure. And really, the combination of all these different projects are answering the key tenet in the center, how vulnerability and resilience factors affect brain functional and structural connectivity. So how are we made up right now? The AABC, as you can see, it consists of multiple projects, which are on the left-hand side. But more importantly, there are multiple cores. And these cores will be feeding into each of the different projects so that the projects can answer those key points that we just discussed. So included in that is the IDAC core that will be led by Drs. Harms, Terpstra, and Jacob, which will be looking at multi-site acquisition using the current sequences that many of us know and love of the HCPA. There'll be a genetics and multi-omics core that will be led by Drs. Kuchaga and Arnold. There'll be an informatics and data analysis and statistics core led by Drs. Glasser, Marcus, and Nichols, as you can see in the bottom. And then there will be, of course, data sharing from any of these cores and projects. As you can see, those projects will then answer particular key points, and there will be an administrative core with 3 of the 4 investigators here today. Now, on the right-hand side, as you can see, the data acquisition or the flow that will be occurring. As I said to you before, there will be 4 sites that will continue to acquire at the same kinds of scanners that they were for the HCPA. That data then will be fed for the different projects, and then at each of those 4 sites, there will be multiple different blood samples that will also be collected, as well as questionnaires, and I will be going through those with you in a minute. And then that data will be fed to the various cores. The cores will then have interactions among themselves, and then provide analyses. And you will be seeing some of the newest stuff by Dr. Glasser, who I'm so sorry he could not be here today. Will show you some of the new data processing cores that will be present for some of the analysis. Well, it really takes a village, as you already heard David talking about. We have a fantastic leadership team. You can see multiple project leaders that are there on the lower level. And then we also have different cores. And as you can see, we have multiple different cores from the administrative, the integrative data acquisition, informatics. And we have also made it a priority to have diversity, recruitment, and retention. You can see two of those members of our team that are doing that. What we've noticed is we've been very fortunate enough to have about 25 to 30% of all of our individuals of underrepresented groups, but we are trying to increase that so that we have a more equitable and more understanding of these key factors in groups that are commonly not studied. And then we also, of course, have the genetics and multi-omics core. So what is the flow? It's going to be very similar to what was done before in the HCP. We'll be doing pre-study analysis, so screening and scheduling. Any individual who was in the HCPA will then be asked to come back for now the AABC, and then we will be recruiting, as you heard, an additional cohort of individuals that will be studied at 2 time points. And those individuals, regardless, on the first day, they'll be getting consenting blood draws, asking questionnaires. There'll be MRI studies as well as NIH toolbox. This is a long day, and so sometimes it can be split, as you can see by the arrow, if it needs to be for 2 sessions, especially for our older adults. And then there'll also be, if they continue to do the whole day, more cognitive testing, scanning, as well as satisfaction surveys. And then there will be additional questions and phone interviews post-study. So this is very similar to what we've seen in the HCPA. So what are the variables that we'll be collecting? It's a long list. You can refer to these, or if you have more questions, please feel free to contact me in those. We have the DSMV being collected even in the pre-screening. Here are all the variables that will be collected during their process when they're in here for the study visit, including blood and plasma, drugs and alcohol questions, biometrics, NIH toolbox. As you can see, a whole laundry list. I'm not going to be reading through it, but it's a long day for our individuals and those who are participating, but we try to give them breaks. The imaging collected is similar to what has been seen before. There'll be both an MR session 1 and MR session 2. You can see in the first session, there'll be resting state with a whole host of other, including functional tasks, go, no-go tasks, and face naming. And then in MR session 2, there will begin more functional connectivity and diffusion, as well as ASL. And then for 2 sites, and you'll be seeing this by Dr. Terpstra, there'll be MRS collection that will be collected at a separate time point. What variables will we also looking at after the individual leaves us? Well, we'll be applying first actigraphy, so simple kinds of ways to measure the number of steps, as well as movement, skin conductance for individuals, so specifically women, for vasomotor symptoms, and then even a food diary as well. So how will this all be integrated? Well, we'll be taking those brain measures and those subject measures. We'll then be kind of compressing them and looking them in either different domains. We'll do dimension reductions, and then you can see we're doing CCA, so canonical correlations, and we'll be looking at this with Dr. Nichols and others to kind of give a weighting system to kind of understand what are those key factors. So what are some of the interesting results that we're already seeing by looking at the HCPA data and that are leading us into the AABC? As you can see on the left-hand side, there's an allostatic load, so measurements for Project 1, as well as perceived stress scores. And you can see that in individuals that are the youngest, and I wish I could be in that group now, but I'm no longer, is that less than 50, we're seeing individuals that have their perceived stress scores are the highest, and then you can see decreases, but that's also related to their allostatic load. And then you can see that with higher allostatic load, in the bottom left-hand side is that there's a higher perceived risk score. So it looks like these stressors are being translated into this allostatic load. And then in our imaging measures, which you will be seeing by Dr. Rashid, is that there are different areas in the default mode network that seem to be affected and related to this allostatic load. And then we've been also looking at Project 3, and you'll be seeing some results also by Tyler Wishard, who will be looking at some of these things. We've looked at menopause, and you can see either pre, peri, or post-menopause, and in relationship to the default mode connectivity. And you can see that post-menopause on the left-hand side, there are decreases in this functional connectivity in women who are postmenopausal compared to premenopausal. And then we also see in the postmenopausal individuals, there's a reduction in their A beta 42 to 40, which is their amyloid load, a marker of Alzheimer's disease. And in those individuals in postmenopausal, there seems to be this reduction. And so the questions of how menopause interrelates with both Alzheimer's disease as well as functional imaging measures will be looked at in Project 3. Well, this wouldn't be able to be done, and as you saw already from Dr. Salat, it's a whole group of individuals. We look very forward to this, and again, like to acknowledge our whole great team that is here, including Angela Oliver, who's our project manager. And I'll be handing off to Dr. Glasser, who will be giving our first talk, but it'll be through video. One last thing, if you have questions, we'll be having something at the end. Please come and use the microphones for that. Strongly suggest that. We'll be also trying to look at the app. We don't know how well it's working, so.
They're working now.
It's now working. We've just been told that, so sorry.
Into this virtual presentation. I regret that I was unable to attend Glasgow due to a couple of important personal events, a close friend's wedding and residency graduation that happened the same weekend. So I'm going to talk about the HB style approach to brain imaging and imaging data phenotypes. So first covering what is HCP style neuroimaging data, how best to pre-process HCP style data to maintain its quality, and then what I mean by imaging data phenotypes. So first, HCP style neuroimaging data. So I mean high quality multimodal MRI data, including structural, functional, and often diffusion MRI acquired on as many subjects as is feasible. High spatial, temporal, and angular resolutions, enabling accurate neuroanatomical mapping, selective fMRI denoising, and maximum achievable accuracy of structural connectivity. And to achieve these goals really requires a 3 Tesla or higher field scanner, a 32 channel or higher head coil with optimized pulse sequences such as a VNAV encoded structural images and multiband EPI. So one of the really key points of HCP style MRI acquisition is choosing spatial resolution based on neuroanatomical considerations. So we have here a group average cortical thickness map and the histogram of the cortical thickness map. And we can see the mean human cortical thickness is around 2.6 millimeters. The minimum thickness is 1.6 millimeters. And so, those fMRI studies with resolution that is coarser than 2.6 millimeters, will turn them low resolution fMRI. High resolution fMRI is those that have better than 2.6 millimeter resolution. Most brain imaging studies are still low resolution fMRI, but that's changing. And ideally, we want to have a resolution, better than mean cortical thickness so that we can accurately represent what's going on. So in the Human Connectome Project and Adult Aging Brain Connectome Project, or AABC, we had 2mm isotropic resolution for our fMRI. For our arterial spin labeling, or ASL data, we were at 2.5mm isotropic resolution. For our structural scans, our T1s and T2s, we ideally want to be at 0.8mm isotropic or less, basically half that minimum cortical thickness of 1.6mm. And we were at 0.8 for the Human Connectome Project Aging and AABC projects, and a little higher for the young adult. And then tractography is a really resolution-starved problem. So diffusion MRI resolution should be maximized to whatever you can do based on the amount of time available in the study and SNR. 1.5mm isotropic resolution for the aging project in AABC, and a little higher for the young adult. So what happens if the resolution is too low? Well, this is a sort of a measure of confusion of where signal is coming from across cortical folds and different fMRI resolutions. And we can see there's quite a bit of this confusion at 4 or 3mm resolutions, but at 2.5, it's decreased quite a bit, and at 2, it's pretty much non-existent. So if our resolution is too low, it can be hard to tell if an activation is on one side or the other of a fold, and the two sides of a fold may be very different in terms of sensory and motor cortex. Other considerations, it really helps to have a VNav, a navigator inside of the 3D T1 weighted and T2 weighted scans, because that can be used to improve image quality by realigning and reacquiring TRs if there's head motion. This reduces the need to rescan the patient or even throw out whole subjects. For fMRI resolution, we really want temporal resolution. We want to have the TR less than 1 second. We were at 0.8 for the Human Connectome Project Aging and AABC projects, a little higher for the young adult. And this enables selective denoising. We got to be using a multi-band factor of 8 to achieve this for 2mm data. And this selective denoising allows us to remove the artifacts without modifying the signal. We want to get as many minutes of fMRI as possible and diffusion directions as is feasible. Using phase reversed images for diffusion data and spin echo phase reversed images that are matched to the fMRI and ASL data is very important. And this can be used for B0 distortion correction and even pseudo B1 mapping. Using multiband EPI for the ASL data enables high isotropic resolution with multiple labeling delays. We had a few lessons learned when we have done all this with the Human Connectome Project, things we wish we had done differently. One of those is it'd be better to have had rescan normalize on for everything. Multi-echo NP-RAGE turned out to be more trouble than it was worth. And at least in our hands, multi-echo fMRI wasn't clearly beneficial either, although I know that's a controversial position. So we've got this high quality data. How do we maintain its quality throughout pre-processing? We really want to minimize blurring induced by preprocessing, for example, from multiple trilinear interpolations or overt smoothing. Remove the spatial effects of head motion within and between modalities. Using BBR for precise cross-modal within subject volume registration really helps with that. We want to remove all the image distortions so that each MRI image accurately represents the physical space of the subject, and then we want to precisely align brains across subjects. And to do that, the HCP proposed a new standard coordinate system that we term SIFTY gray ordinates. And so the gray ordinate standard space is a combination of surface vertices, 30,000 in the left hemisphere, 30,000 in the right hemisphere, and 30,000 subcortical voxels. And this allows us to use surface meshes for aligning the sheet-like cortical structures and nonlinear volume registration for aligning non-cortical structures. And in terms of this gray ordinate space, we have around 90,000 gray ordinates with 2mm average vertex spacing and 2mm subcortical voxels. So how do we accurately align cortical areas across subjects? Well, we can take area MT as an example. And it turns out a volume-based registration does pretty bad for area MT. We've got 2 different hotspots of relatively low probability, or maximum overlap isn't even 50%. And it's kind of a blurry mess here. Folding-based registration on the surface does quite a bit better. We got a single hotspot of high probability, but our overlap still isn't all that much above 50%. And we've got number of individual area MTs and individual subjects that are not lined up. But if we do what's called an aerial feature-based registration, we can get up to 100% overlap, getting all of the area MTs lined up. And by aerial features, I mean things like architecture, function, connectivity, topography, or the areas themselves. We talked about imaging with a fast TR, and that really helps to remove artifacts and noise from the fMRI data. So we can take a functional connectivity map here. So here's the seed location with this white sphere. Here's a cartoon time series, and this basic functional connectivity map is the correlation value of this time series for all of the other time series in the brain here, and this is without any data cleanup. And doing these type of correlational analyses are really vulnerable to any kinds of non-random artifacts in the data. They produce biases. This doesn't really look like much if in terms of a neurobiological effect, but if we use spatial ICA to remove the effects of head motion, all of a sudden now we can start to see the default mode network popping out here. If we use temporal ICA to remove the effects of subject respiration, now the default mode network is made more specific. We can see there are semantic correlations. And then if we remove random measurement noise, for example, with Wishart filtering, this boosts the correlation values quite a bit and makes a nice default mode network from this single surface vertex seed here. So most connectivity studies try to remove artifacts, but it's really important to remove noise without changing the neural fluctuations of interest. And activation studies benefit as well. So you can remove biases and improve statistics by doing these types of corrections. And again, hitting these targets of temporal and spatial resolution really require multi-band EPI. So we've got our data pre-processed. What about these imaging data phenotypes? Well, what do I mean by that? This is something similar to the imaging-derived phenotypes, which is something that somebody else came up with. And I happen to forget what the D stood for. So I've made a IDPs that's a slightly different acronym, and I'm going to have a slightly different take on it than they did in the biobank paper. But the basic, my take on this is using the brain's structural and functional neuroanatomy to partition measures of interest in a neurobiologically meaningful way. So that means using things like brain areas or functional networks or white matter tracks and averaging within these neuroanatomical units to reduce the effects of random noise, improving one's statistical sensitivity, and also collapsing the voxel, vertex-wise, or gradient-wise statistics in these neuroanatomical units to reduce the number of statistical tests, which improves one's statistical power. So as an example of cortical areas, we have the Human Connectome Project's multimodal cortical parcellation, where we've got auditory areas in red, sensory motor in green, visual in blue, and default mode or task negative in the darker colors, or anti-default mode and task positive in the brighter colors, with mixed coloration, for example, here in LIPV, when we have mixing of both sensory motor and visual information. So how does this work? Well, we've got our aerial borders and we've got a myelin map where our high myelins are in the reds, yellows, and oranges, and the low myelins, the blacks, purples, and blues. And this is around 50,000 data points that we see here. But we can average myelin content within each cortical area and produce a map that looks like this. And this is now only 360 data points, as there are 180 cortical areas per hemisphere. And if we just flip between these two, we can see that the pattern is really quite similar. We haven't lost very much information despite decreasing the size of our data, the number of statistical tests enormously by 2 orders of magnitude. You can do a parcellated analysis with functional connectivity. Here's a full correlation analysis on the left inferior parietal cortex showing the default mode network. Partial correlation is a sparser analysis, but it still shows the default mode network very nicely. You can do this with task fMRI. Here we have working memory contrast. This is a language contrast. And if we compare the parcellated analysis and just a regular gradient-wise or vertex-wise analysis that we have here, they're scaled with the same z-statistic, you can see that the results of the parcellated analyses are clearly more significant than those of the dense analysis. And in fact, if you just compare, averaging, the z-stats within the areas versus averaging the time series and then fitting the GLM, if those things were equivalent, we'd have all the blue dots along this red line here. But it turns out that if you make those average time series before fitting the GLM, you get quite a bit more significant statistics as these dots are steeper. Functional networks are another approach that one can use, and you can consider either hard definitions versus spatial weighted overlapping ones. One hard definition of functional networks was proposed by G et al. Here. Each area or each brain vertex, voxel, or gray ordinate typically has been assigned only one and only one functional network. But that's not the only way to do it. You can use a weighted approach to define functional networks. In this case, we've found the main default mode component across 5 different paradigms, including 7T and 3T resting state data, traditional tasks at 3T, a movie task at 7T, and retinotopic data at 7T. Another approach that makes weighted functionally overlapping networks, temporal ICA is done at the group. Probabilistic functional modes, or PFMs, are done in both at the group and individual levels at the same time. And also the probabilistic functional modes don't enforce orthogonality like the temporal ICA. Things must be temporally orthogonal. And these type of approaches, in this case, temporal ICA, can do a great job of pulling out interesting things. So here's a weighted functional network pulled out by temporal ICA, and here's the traditional task analysis, basically showing the same thing in this 3T task data. And each cortical area most likely participates in multiple spatially overlapping functional networks generating different specific behaviors. So this idea of a one functional network per area per pointed space is probably an oversimplification. All right, so just to conclude, what kind of data is going to be produced for the Adult Aging Brain Connectome project? What are the IDPs that we're making? Well, we've got some at the cortical area level, network level, and the tract level. And we can consider things like the size or the surface area of these, or the volume, these different structures. We can measure architectural information like myelin or cortical thickness. Functional connectivity could be done either area to area or network to network. Structural connectivity can be area to area or based on the tracks. We can do either task activation in a traditional task, or we can just look at the amplitude of the bold fluctuations, either at the areal level or network level. Blood flow can be measured in areas, networks, or tracks, and we can look at white matter hyperdensities as well. So to summarize, we want to get high quality MRI data that enables accurate mapping to the brain's structural and functional neuroanatomy. We want to pre-process this data to preserve its high data quality and enable localization of these measures to this neuroanatomy. We want to use the brain's neuroanatomy to subdivide the data to improve both statistical sensitivity and power. And this HCP style approach is really helpful in addressing the increased focus on rigor and reproducibility of neuroimaging results that's been seen in the field. Well, there are many people in the Human Connectome Project, and the Young Adult Project, the Aging Project, and the AABC to acknowledge who I've listed here, and I may have missed others. And I thank you for your attention.
Good morning. I'm Borna Lee, and before I proceed, I would like to thank the organizers for inviting us to present some of our recent findings from the Human Connectome Project in Aging, or the HCA study. From the previous presentations, you have heard about the structure and the methods of the HCA study. Now I will provide some examples of applications to answer our lifespan questions about risk and resilience. So we are primarily interested in the factors that contribute to risk, for example, health factors that are known to contribute to impairment in late life, and neural or connectivity mechanisms of resilience, for example, what promotes high functioning in late life. So we wanted to explore how cardiovascular and metabolic risk factors affect functional connectivity across the lifespan. To do that, we used data from 930 participants acquired in HCA to assess the impact of cardiovascular and metabolic risk on the whole brain functional connectivity. And for that, we used resting state fMRI data processed by the data core, which was discussed by Dr. Matt Glasser in the previous presentation. And the age range for this sample is between 36 and 90 plus years, with about like 55% women in the sample. So to assess cardiovascular and metabolic risk factors, we used a composite index of cardiovascular and metabolic, or CVM health factors, of averaged Z-transformed measures. And this composite measure included 12 factors encompassing vascular, metabolic, and immune systems that are known to increase risk for or correlate with CVM health. Some of the risk factors we used included blood pressure, BMI, and cholesterol. And note that a higher value of CVMI indicates poorer cumulative CVM health. So typically, when we examine the association between brain connectivity and health or any other factors, for example, in this case, cardiovascular and metabolic index, or the CVMI, we can get two different types of association, which are positive and negative associations. And in positive association, the direction of relationship between connectivity and cardiovascular risk is positive, whereas in the negative association, this direction of relationship is negative. But this does not tell us whether the correlation is getting stronger or weaker. And it just tells us the direction of the change, but an effect can have a positive slope, and then it can either weaken a correlation or strengthen a correlation. In addition, the whole brain connectivity effects are typically visualized as heat maps. But if we could project these whole brain effects onto the brain surface, that might be more helpful in terms of interpretation, as well as finding hotspots of like greater effects. So in this study, more than 98% of the significant associations between connectivity and cardiovascular risk were weakening associations. Therefore, I will mostly focus on explaining the conceptual background that are relevant to the weakening association. So if the association and the mean connectivity show opposite signs, then we define it as a weakening association. For example, in this part A, the mean connectivity is positive, while the association is negative. Similarly, in part B on the right side, the mean connectivity is negative, while the association is positive. So in other words, a weakening effect is when the strength of the correlation or the anticorrelation is decreasing with increasing risk. So we test the association between the risk factors and the pairwise correlation between every region and every other region within the brain. Then we sum them up as a summary measure. For the brain regions, we use the Glasser 2016 parcellation as discussed in the previous presentation. Okay. The cartoon figure in part A demonstrates our approach for estimating the summary maps. Here, first we find out whether the association between the CVMI and a pairwise correlation between a particular brain region with every other region is significant. If the association is significant, then we assign a weight of 1 to this pair. Otherwise, we assign a weight of 0. So, for example, in this figure, you can see we had 2 significant associations. Therefore, we assigned a weight of 2 for that region. So finally, we sum these weights together across each region, and then we find the summary measure, which is the degree of strengthening or weakening for that region. And for correction for multiple comparisons, we used a permutation test, and then we applied FDR correction. And before we move on to the results, let's take a look at an example of the weakening effects of the summary map. So here, the darker blue indicates lower weakening degree, whereas a lighter green color indicates a higher weakening degree. For example, this left posterior insular region showed a CVMI degree of 275, which means that in this case, when we test, there is a significant association between the left posterior insular region and 275 other paired region after regressing out the cardiovascular risk measure. And these are the results showing the CVMI-related weakening degrees. And just to remind you, the darker blue indicates lower weakening degrees, whereas lighter green indicates a higher weakening degree. So first, we looked into the whole brain weakening degree without any adjustment for age. And these results show a global weakening effect, which is summarizing the total number of connections across each brain region that showed a decrease in connectivity with increased risk. So after adjusting for age, while the total number of significant associations decreased, we can still see similar patterns of weakening across the brain, showing a larger effect. We also found strong bilateral symmetry in weakening effects with and without adjusting for age. And some of the notable regions with more weakening degrees include regions in the frontal and lateral parietal cortex, as well as several regions in the medial prefrontal and cingulate cortex. And note that here, any region showing a color, these are the regions that showed a significant association after the permutation testing and FDR correction. And I would also like to note that in this context, strengthening or weakening does not necessarily indicate the functional integrity. And we also wanted to examine how weakening effects are distributed across different age groups. So, for example, we wanted to know if the effects are replicated across middle-aged and older groups, or if one group is more affected than the other group. So the younger and middle-aged group is defined using the participants below 65 years, while the older group is defined using the participants who are 65 plus and above. And from this figure, you can see that while the effects are fairly replicated across the brain, the younger and middle-aged group is showing relatively greater weakening effect compared to the older group. Also, the older group is showing greater weakening effect in the primary sensory area, as you can see from the figure on the right side, whereas the younger and middle-aged group is showing greater weakening effect in the limbic area. Finally, we also wanted to know how weakening effects appear in the 80 plus or the oldest old sample. And while we had a much smaller sample size for this group, we can still see some patterns of weakening in the temporal lobe similar to the other age groups. And we also used a cognitive composite index to first identify high and low performers based on the top and bottom 50% of the sample across the age regression line, as you can see from the top part of the figure. And we found that overall the low performers have higher cardiovascular risk than the high performers across the age. And in future, we will look into how brain connectivity, cognition, and risk are all related to each other. And to summarize, cardiovascular and metabolic risk effects are stronger in younger and middle-aged adults compared to the older adults, which highlights the need for early attention to cardiovascular and metabolic factors in adults for optimal brain health and overall functioning. And this work would not be possible without the contribution from these individuals listed on this slide. In addition, we would like to acknowledge the support from National Institutes of Health, including the National Institute on Aging for the current study. And thank you all for listening to my presentation. Next, Dr. Melissa Terpstra will give the final presentation in which she will discuss novel magnetic resonance spectroscopy applications to the Human Connectome project.
Thank you, organizers, for inviting our symposium, and thank you, colleagues, for so nicely describing what this study is all about. Now I'm going to tell you about how and why we're adding 7 Tesla spectroscopy to the study. So back when over the last 5 years, when we've been running the Human Connectome project across the consortium, I added a separate R01 to do 7 Tesla spectroscopy on Minnesota's cohort. So now when we go into the next phase, as was described, spectroscopy will become part of the whole project. It'll all be in one grant. So as a really quick outline, I'm going to start out assuming that many of you don't know a lot about spectroscopy and summarize 7 Tesla spectroscopy and how it works. Then I'm going to talk to you about how I've created MRS-based classifiers of aging based on my published data, and how I'm going to apply that to study correlations with cognition and genetic risk from the Connectome cohort. I'll talk briefly about what we think maybe some of these signals mean, so how there's a study on myo-inositol and PET measures of inflammation. And finally, I'm going to show you our progress with enfolding spectroscopy into the Connectome. So first, as I promised, a brief primer on 7 Tesla spectroscopy. So we're using ultra-high field, ultra-short echo time spectroscopy. The short echo time is important in order to avoid T2-related confounds on the aging and Alzheimer's disease data. So on the left here, you have a typical human brain in vivo spectrum from 7 Tesla on the top. You can see the typical NAA, creatine, and choline resonances that are commonly seen at lower field, but you can also see that many, many more resonances are resolved. Similar to other methods, we use LC model to fit our spectra. So what that is we know what chemicals we expect to be present in our spectrum. So the basis spectrum on the bottom left are basis spectra that we feed into the model, and then the modeling decides how much of each chemical is in that spectrum. So at higher field, the frequency resolution is better, and we're able to see more chemicals. You can see the ASC symbol and the GSH. Those are antioxidants, and they're important to my research in aging and Alzheimer's disease. So you can see how very small and highly overlapped those signals are. They have several bumps on the line, so they have a unique signature, and so therefore we are actually able to see them. On the right is a summary table of my published paper in Alzheimer's disease, where you're able to see that we can measure 14 chemicals using this methodology. And so we were able to replicate the findings in total choline and myo-inositol that have been found at lower field, but now we're able to find changes, for example, in ascorbate that we had not been able to see before. if you have a hard time believing that we can actually measure those tiny signals in that highly overlapped spectrum, here are a few studies that I have done to validate this. So first of all, on the left, my pharmacy collaborators in Minnesota approached me and asked me if I would be able to measure a brain change in glutathione signal based on the drug N-acetylcysteine, which is a precursor for cysteine, which is a rate-limiting step for glutathione production. So they wanted to give N-acetylcysteine to the participants intravenously and watch what happens in the brain. So we measured the N-acetylcysteine at time point 0 on the far left here. From the pharmacokinetic modeling, we predicted that the highest brain glutathione would occur in the range 1 to 2 hours. Participant couldn't stay in the scanner that long, so they came out of the scanner, and then they went back in for the last part. On the bottom, you can see the blood redox ratio. So we were able, the NAC arrived in the blood at about time point 1 hour. Glutathione peaked at more like 2 hours. So you can see the spectra are very high quality at both time points, and they look exactly the same. That's really the point. Very high quality, but you can't see the signal, but obviously we are able to measure it. On the right, you see a similar study, but it's done more theoretically. So the spectrum on the far right was measured from the human brain. We did 5 of these with retests, and we compared to 5 other people to get different noise realizations. But what we did was we were able to fit the native glutathione concentration, and then we took measured phantom data, and we added and subtracted known amounts of glutathione signal, plus and minus 100%. We put the altered spectra back through the model and verified that we were able to measure these tiny changes in glutathione that we expected. And interestingly, you can see at minus 100%, the model accurately measures that there is no glutathione there. So the model does not make up something that's not actually present. So now to go back to tell you about how I trained classifiers for aging and Alzheimer's disease using these spectroscopy data. So in this study, I'm going to focus on the posterior cingulate cortex. It's an important brain region for Alzheimer's disease because it has plaque and tangle pathology, changes in glucose metabolism, changes in blood flow, and changes in connectivity. So this is the region I'm focusing on in this talk. So in the published paper on aging, we had 16 young and 33 older participants. The older participants were seen by my neurologist colleague to make sure that they could not possibly have any signs of incipient Alzheimer's disease. And so in the aging study, those older adults also had to have a MoCA score greater than 24. So in the figure where you see older adults versus young, we found several age-associated differences in the brain neurochemical profile. Similarly, we did a study on Alzheimer's disease. So once again, the neurologist and actually a neuropsychologist collaborators were involved at the VA Medical Center, and they did consensus diagnosis to refer me early stage participants with early stage Alzheimer's disease. And you can see on the far right that we found the expected and well-reported changes that have been seen with 3 Tesla spectroscopy in total choline and myoinositol. But using this methodology, we were able to also measure a higher ascorbate signal in participants with Alzheimer's disease. So on the bottom, if you look at the random forest analysis that are also published in that paper, we found that using this neurochemical profile, we were able to distinguish patients from controls with same sample sensitivity of 88% and specificity 97%. And the main predictors in that model were indeed ascorbate and myoinositol, with ascorbate being the most important predictor. So now we go to how I've added spectroscopy to the Connectome study. So on the left is a heat map that we generated way back from lifespan data in order to find the hottest regions of default mode connectivity, and that is where we placed our spectroscopy voxels. I'm going to focus on the posterior cingulate cortex. So now in the Connectome, it's been well described by my colleagues what the study is all about. In my sub-study, in the grant that I got to add spectroscopy to the Connectome, it was an Alzheimer's disease-focused grant, so I had to include only participants who passed screening by my neurologist and my neuropsychologist as having no possible incipient Alzheimer's disease. So that's a bit of a subset of the typical aging cohort. I have to thank my co-PI who's handling some special analysis of these voxel-based connectivity measures, and of course my co-PIs at the University of Minnesota who helped me become part of this project and have this opportunity, and the neurologist, neuropsychologist at the VA. So on the left is some of the preliminary data that is really exciting and part of why we added this to the connectome. So we chose 5 focused neurochemicals for this grant. Some of them are easier to see at lower field and some of them very hard to see at lower field. And you can see in summary that there's an interesting inflection point in the trajectory of the brain chemistry at around age 50. On the right is something I haven't told you about yet, but we're actually, because this was an Alzheimer's focused grant, we are measuring PET scanning on all of the people who are 60 years old and older. So I have a neurologist at University of Minnesota reading these as PET negative or PET positive. And as most of you probably know, that there's not a one-to-one correspondence between the cognitive symptoms of what is called probable Alzheimer's disease compared to the pathological manifestation of Alzheimer's disease. So about 30% of people who are cognitively healthy are known to actually have amyloid pathology in their brain. So it's not a surprise in my sample that that's also the case. So on the graph on the right, you see a plot of the concentration of brain NAA. N-acetylcysteine is thought to be a marker of neuronal integrity, and so we have actually higher NAA concentration in our participants who are PET positive, and we think this is one of the things that suggests that resiliency could be playing a role here, meaning that their healthy brain chemistry might be protecting them from the negative effects of their pathology. So this is the heart of my talk. This is the published data. So on the left, this is a principal component analysis. So we did everything we could to make sure the model was not informed at all about aging or Alzheimer's disease status. And we just threw this in a completely untrained principal component analysis. And you can see on the left that aging was separated with 100% perfect separation just on one principal component. On the right, the Alzheimer's disease groups were not as well separated, but decently separated on 2 principal components. As a sanity check, what we did now is we just used the formulas that came from the principal components, and we were able to plot what the Alzheimer's participants look like in the aging space. And you can see some of them overlap with normal aging, and some of them seem to be in their own group. Similarly, the young people are in their own space and not anywhere near looking like Alzheimer's disease. But now, let's add the connectome cohort data to this. So the training data were binary, so young and old people, whereas the connectome now has the age span between. Nicely, you can see, of course, we didn't end up including anybody with Alzheimer's disease. None of our new cohort is in the red dot space on the left. There is an interesting participant here who, though they could not possibly have been younger than age 35, have a fairly young-looking brain chemistry. Similarly, we plotted the connectome participants in this space, and they kind of fall with a similar story. But what I want you to remember from this is if you look towards the left on any grant, on either graph, towards the left is either older or more Alzheimer-like. And I'll carry this through to the next slide. So what we did next was with my colleagues, as we wrote the new U19 grant, Tom Nichols created a cognitive composite score from the connectome data, and he created this composite in both the memory and executive domain. And also Carlos Cruchaga took the blood samples and measured the polygenic risk for Alzheimer's disease, and as is common to that field, that can be done with or without ApoE4 in status in the model. And we asked the question, what among brain chemistry, cognition, and polygenic risk are most correlated with one another? And we found by far that the brain chemistry and the cognitive scores are the most correlated. Carlos is not in the least bit surprised about this because polygenic risk is something you're born with, whereas your brain chemistry and your cognitive function are more related to what goes on in your entire life. So I also just wanted to point out that as I'm plotting the executive function, the strongest correlation on the right here. So remember, as you go towards the left, you're either older looking or you're more Alzheimer's-like. And indeed, as you're moving more toward the left, you're also having a poorer cognitive function. This I would like to highlight that now that we're going into the next project, we're going to also be doing spectroscopy at the MGH, and this is a paper by my colleague there, Eva Ratai, who is running that effort at MGH in spectroscopy at 7 Tesla. So to be very brief, this is a paper in patients with ASL and ALS, sorry, and they, what she has here is a PET tracer for activated glia. So what she did was she measured the MRS spectroscopy myoinositol signal, and she also did PET scanning, and it turns out that only in the brain regions that have pathology with this disease, then you find a relationship between these two markers, myoinositol and PET, marker of glial activation. Now, neither measure is a pure and specific marker for just glial activation, but they're supporting one another, suggesting that we're measuring features of Alzheimer's disease, of inflammation. I also think that the ascorbate marker is probably related to chronically activated microglia, but there's a long ways to go to figure out exactly what it really means. So finally, here's where we're at with adding spectroscopy to the connectome. So on the top, on the left, you see spectra that we actually had in the grant. So when we wrote it, we made sure that the high spectral quality that we've been getting all along was reproduced using the 7 Tesla scanner at the MGH with the NOVA 1x32 Siemens provided coil. The map at the top was provided by Matt, illustrating that now we're going to be able to match up what we're measuring with spectroscopy with the kind of all of the outcome measures that are coming from the connectome. The image on the right was taken at the University of Missouri, where I moved to, and as a shout out to our Siemens partnership there, we were very quickly, within weeks, able to measure exactly the same spectral quality there. And this is the MP2 RAGE image that Isa Yakub created the parameters for in Minnesota on the Terra there, and we're getting really nice images in this project as well. So you might be interested in that. We're spending about 8.5 minutes on really good images on in this study as well. And so this is a segmentation that was done by my colleagues in Missouri. They really had very little trouble with this. And finally, in case I forgot to acknowledge any of the people in the project throughout my talk, here they are. And also, of course, the funding that drives all of this project and allowed me to add spectroscopy to this project.
Thank you, Melissa, and thank you all for being here. You can ask questions on the app, or also you can come up to the microphones. So we have some questions already. I'm going to start with a question for Bo, and maybe David will weigh in. Is resilience connected in any way with cognitive reserve? Do you believe it will be able to quantify and describe better cognitive reserve with this project?
So that's a great question, and I'll start, but we also have Dr. Salat, who's an expert on this as well, too. So the idea is that, yes, cognitive reserve is providing us, but many of you know, we kind of use education as one of our proxies for understanding cognitive reserve and using that as a measure for resilience. What you've been hearing today already by Dr. Rashid and others, and we've highlighted in the talk, is that there are multiple other factors that we'll be measuring in this. You heard about allostatic load, you heard about cardiovascular measures that are also going to be very important factors. So those are individuals who have lower cardiovascular risk, or those individuals who have other perceived stress measures that are reduced, or it may also be their environment, and we'll be capturing that through something called the Area Deprivation Index. And by kind of capturing those kinds of measures, we'll really start to understand who are individuals at risk or who are individuals who have increase in resilience. So I think that this will be better ways to describe this than are currently being done, but I'll throw it to David.
Yeah, I'll just add to that briefly. I think that, I think in terms of cognitive reserve, we have to think of what's going on both functionally and what's going on in the brain, and the sorts of measures that we're acquiring here will hopefully allow us to do that. Reserve is often, considered protective, but at the same time, it may not be protective against what's going on in the brain. The degeneration may still be occurring, but people may be less expressive of symptoms for cognitive reserve. There may also be neural reserve, where people have some brain mechanisms that protect them. And then there are, of course, all these additional factors that may be both modifying both cognitive and neural reserve. So I think we'll be able to look at those measures in these studies.
Thank you. There's a question about multi-echo, MP RAGE, and fMRI, and I think that was an error, and maybe David can explain that. The MP RAGE is multi-echo. It's the fMRI that's single echo, correct? Okay. Bo, do you want to say something real quick about the DSM-4 and why we use that?
Yeah, we use the DSM-4 because that was how it was acquired previously, and so that it allows us to continue to move forward in that. The individual also asked about, DSM-5 criteria that is now more established, and we will be getting some of those kinds of measurements, but 4 has been the way we allow us to compare to previous studies.
For Melissa, is the MRS data going to be openly available as part of the HCP?
Oh, I'm
She
sorry.
needs a mic.
So in the AABC, that will be publicly available through the cores, and from the past data, that will follow the typical NIH rules. So at the end of the grant reporting period, we will be compiling those data, making them available.
Great. And to follow up on the MRS, there was a question about what happened where MRS and PET did not overlap for neuroinflammation.
Can you say that MRS did not?
Yeah, what happened in places where MRS, just as what happened where MRS and PET did not overlap for neuroinflammation?
So that's Ava's paper, cases where MRS.
It sounded like some places they overlapped and some places they not.
Yeah, so what I remember from the paper is there were the brain regions where there was overlap were the brain regions that were most that are most implicated in ALS, and that's where you expect the inflammation to be. And where there were not overlap were brain regions that are not involved in that disease. And also, she did do some correlations of myoinositol with rating scores in ALS, so the severity of the disease, and those also correlated with myoinositol concentrations. Does that answer the question?
I think so. There was a question about remote digital data, and the answer is we did not collect it in the original HCPA, but we will be collecting a week of actigraphy data on everybody in AABC. And in addition, for women in the perimenopausal years, for the menopause project, we'll also be collecting vasomotor symptom data, also longitudinally with digital wearables. So, and that will all obviously be publicly available. This is for Barnley. How did you factor in variability of CRP into CVMI to get an overall inflammatory measurement, as it's highly variable?
So for the composite, we only considered taking the z-score of each of these variables, but we did not remove any separate variance from the C-reactive protein.
Okay, great, thank you. So there are lots of other questions here. I don't know if we'll be able to get to all of them. Melissa, in regards to the high field MRS, are you able to tell the difference between different tau proteins? And maybe Bo can weigh in on that as well.
The ability to measure tau with MRS?
Yes.
So we haven't done any measurements of tau in the study so far. Are you just asking me to clarify that, Bo actually picked this up? There's taurine signals that we measure, not tau signal itself with spectroscopy. So we can't pick up signal from tau or amyloid with spectroscopy directly.
Okay. And we have some tau measures that Bo can talk
Yeah,
about.
so we will, if the question's in regards to how that will correlate, so the MRS signals that you already heard from Melissa, we will be collecting plasma measures, and you heard about amyloid measures. We'll also be getting tau measurements, and we'll actually be getting multiple tau phosphorylation sites with Dr. Arnold. So we will be having that ability to kind of correlate that with some of the MRS measures in those individuals who have both.
Okay. There's a question about availability. Is the new data set already available for researchers, and are there cognitive scores available to assess normal brain function? So the data from the first 5 years of HCPA are in various states of availability now, and that includes all of the cognitive data. And during recruitment, of course, all of our subjects were normal volunteers. We did not directly recruit individuals with Alzheimer's disease for this project, but we're following them a total of 10 years, so some of them will start developing Alzheimer's-like problems. The data, we're just starting to collect the second 5 years of data, so no, they're not available currently. Only the original 5 years are available. The rest will become available as it's collected, but it'll be a few years because we're just getting started. Bo, did you want to mention something about the HIV?
Yeah, so there was a question of the thoughts about an HIV lifespan. Thank you to whoever that is. My other hats of wearing stuff of seeing individuals with HIV, but I would take this in a more larger perspective. What you now will be having access to, and that what the HCPA really allows us to look at, is what is that healthy aging. And so if you were to pick any of your neurodegenerative diseases, and as a neurologist who sees many of these different kinds of patients, you'll be able to then map out if you were doing the HCP-like sequencing, to a known, well-characterized data set. And now with these additional plasma markers, you'll start to learn if some individuals even may have preclinical Alzheimer's disease. And so really this data set will kind of serve as this well-characterized group that then you may be able to characterize and compare to, similar to, and hopefully will be kind of being used to, is those other connectomes that you heard about that I kind of quickly mentioned. So I think this will be a great resource for comparing and contrasting in multiple different diseases.
Great, thank you. There was a question about spatial resolution. Is there similar spatial resolution with 70 MR spec to 70 MRI?
Definitely no. So the single voxel spectroscopy that I'm doing is a large voxel that we're pre-selecting, and that's really a matter of how we get all of the 14 brain chemicals. We do have a pretty big initiative going on in Missouri to get a smaller chemical shift imaging grid, with getting very good instrumentation for that. So it's pretty well known that the spectroscopy is just simply a smaller signal, and you're going to have a smaller resolution, spatial resolution with it.
Yeah, there's a related MRS question about why you measured 2D MRSI to acquire spatial information. Also, why not including a GSH-edited MRS acquisition?
We did not do 2D MRSI in the data that I showed, and we did measure glutathione in all of the profiles. So when I say ascorbate was there, that was one of the 14 that was there, but glutathione was part of the model. So maybe the confusion came from the fact that ascorbate seems to be one of the chemicals that is leading the correlations, but glutathione is in the model. It just wasn't one of the strongest predictors.
And do you plan on correlating structural data with MRS, and what do you expect to find?
So the grant actually was written to answer the question of what leads what. Does brain chemistry lead changes in the structure and function, or does it go the opposite direction? And so my colleague will be creating special ways to analyze voxel-specific connectivity measures, and she's working on that right now.
Thank you. And this is a technical question for Barnley, but is the blue-green color map that you showed perceptually uniform?
So if you were referring to the color maps that's offered by Matplotlib, like the plasma color maps that are perceptually uniform, then no, it's not like exactly like that, because this is something we made using FreeSurfer and we customized it. So the main difference, I would say, is we don't have any smoothing factor here. Like, we don't have any smoothing arch, like going from one part to the next one. I hope that.
Yeah, thank you. And I'm not sure who can answer this question. If Tom Nichols were here, he would. Can we use spatially overlapping regions in models that assume statistical independence of features? That's probably a no, but I don't have Tom here. Yeah, Tom or a Matt question, sorry. But Tom Nichols is floating around, so you should be able to find him at some point. So that's the end of our time, and I'd like to thank all of our speakers and all of you for sitting with us through the end. Thanks very much, and look forward to showing you more data as it is acquired. Thank you.